Hey, hi everyone. I hope all of you had a nice long weekend. And I hope also that the fact that we have an exam this week on Wednesday coming up is not something that's a shock to you, right? I know I'm sure all of you feel like, oh boy, I wish we could go back to having the long weekend. Um, so yeah, we have our next exam coming up on Wednesday. So today we will have our review session for it. So as a reminder, the exam, just like the first exam, is entirely done on La Lima at class time on Wednesday. So at 12:30 normal class time on Wednesday, you don't bother logging in Zoom, right? Because you don't need Zoom. You just need to log on to La Lima, and the exam too will be there at 12:30. It will open up at 12:30, which means before then, I think it does not even show up, right? So don't be confused if you log on at like 12:25 or something and you don't see it, right? And at 12:30, you just refresh your screen and I think it will appear. So it's under tests and quizzes, and it'll just be like the first exam. It's all multiple choice, 50 questions. You have 50 minutes from the time that you click begin, right? So the whole exam is open for an hour from 12:30 to 1:30. And so it's up to you to log in and start it whenever you're ready, keeping in mind that once you start it, you have 50 minutes. So the latest you can start is 12:40 and still have the full 50 minutes before the exam then closes, right, at 1:30. And so, the strategies that you used for studying for the first exam, if they served you well, then I think you'll be in good shape. For those of you who I think I've spent some time meeting with in office hours and things like that since the first exam, hopefully you have a better strategy now for taking it. Okay, so let's go ahead and review what's going to be on it. And so again, of course, in this review session, there's no way I can go over everything in detail, but hopefully this will serve to provide you a pretty good in-depth outline of what you need to review and get ready for Wednesday. All right. So the exam's on chapters 6 through 11, so quite a bit of material. So let's start off with chapter 6, which had to do with the development of B cells. So remember that B cells, just like the other cells of our immune system and our blood, start off as stem cells in our red bone marrow. And so remember, this table here takes you through the different sort of phases or stages of a B cell becoming a B cell. So it first starts off as a stem cell, that hematopoietic stem cell in your bone marrow, and then it will proceed through different stages of development. The pro-B cell stage, during which it's doing VDJ recombination, and the B cell is trying to successfully make a rearrangement of its Ig heavy chain locus, putting together different gene fragments using the oven mitt, right, to put them together. And hopefully it will successfully make a good rearrangement of these gene fragments so that when the cell transcribes and translates a product from that recombined gene, that it is successful in making the Ig heavy chain, right? At that point, then it will go ahead and start making the light chain, right? So that will then move to the pre-B cell stage. And similarly, the oven mitt goes to work and tries to rearrange the different fragments at the light chain loci. And if that works, then you have your finished immunoglobulin molecule being made, and therefore you have an immature B cell that's made. Remember, this table, it is a little bit more complicated than it really has to be, because the reality is what it's just trying to remind you is the status of the heavy chain gene locus, the light chain gene loci, and then the Ig status, whether or not you actually have immunoglobulin proteins detectable on the cell, at that particular stage during the progression. All right, so go back, review the different steps and some of the things like other proteins that are involved in helping to make sure that there's quality control of this process. Okay. We also learned that once you have an immature B cell, remember, immature means, yes, this B cell now has its surface covered in immunoglobulin, the immunoglobulin that it sort of came up with as part of doing VDJ recombination, right? That's the whole cookie recipe thing. So it has its own unique immunoglobulin on its surface with its own unique antigen binding affinity arms on it. But it's immature, right? Because at this point, it's really crucial that the B cell undergoes tolerance training, right, to make sure that the random shaped immunoglobulin that it came up with doesn't end up binding to perfectly normal three-dimensional shapes that are inside the body. Right? After all, the IG was made just through this random process of VDJ recombination. There actually is a really good chance that the random shape that it came up with would actually end up binding something, found in our body, right? There's a multitude of different possible shaped molecules and things that the B cell might encounter when it goes to work. So before it ever leaves the bone marrow, it undergoes central tolerance training. So cells and tissue in the bone marrow are presenting all kinds of interesting potential antigens to the new B cell. Remember, the first IG that it makes on its surface is IgM, right? And if it turns out that that B cell ends up auto-reacting against something like these little gray triangles that are on your perfectly normal, healthy bone marrow cell, then, oh boy, that's bad, right? You don't want this B cell to be let loose in your blood. If this B cell can still have more, if it still has V and J gene fragments left at its light chain loci, like it didn't actually use up all of these gene fragments when it was trying to come up with a productive rearrangement, then it can still try again. It can kind of tweak this, try to do the VJ recombination again, and maybe, right, maybe the shape will be different enough that it doesn't auto-react. But if it cannot succeed in coming up with a IG that does not auto-react, then that's the end of the line for that B cell. It's kind of sad, right? Went through all that trouble just to end up dying in the bone marrow and never be released. And remember, it turns out that the majority of B cells, that's the fate because of the nature of this random coming up with different IG shapes. But if it manages to have an IgM that does not auto-react in the bone marrow, it passes central tolerance training, and now it's let loose in the blood. Now, in the blood, it's still immature, right? Because now it has a chance to interact potentially with even more different normal antigens that are present in your body, right? So it's a very good chance that in the blood, it ends up binding, like, for example, these little red diamonds are just meant to be some totally normal molecule that's floating around in your blood. And if that's the case, oh boy, that's bad, because the problem is this B cell is away from the bone marrow now, right? It can no longer do any more tweaking, VJ recombination. So as a result, this B cell ends up going anergic. Basically, it shuts down in the blood. It doesn't die right away. It just shuts down and nothing else happens to it. And eventually it just gets turned over, right? Maybe after a few weeks, it just naturally dies and gets recycled. We said up until now it's immature because the molecular wiring that's meant to connect the immunoglobulins on its outside with things that are happening inside the nucleus, that chain of different like proteins that need to talk to each other, right, when something binds to the immunoglobulin, it's kind of wired backwards if you want to think about it, right? Because at this point, if something binds to the B cell, it just goes anergic. It doesn't activate. Once the B cell gets to the secondary lymphoid tissue, like, for example, the lymph nodes, right, then it has successfully shown the immune system, not only will it not get into trouble against normal self signals, but it also knows how to exit the blood and go into the lymph system. And at that point, that B cell is rewarded by being made mature, right? So in other words, the wiring of its immunoglobulins on the surface, right? Different proteins get expressed inside of the cell. So now the connection between the receptors and the nucleus in terms of what proteins can talk to each other now will activate the cell if that cell then binds to something on its immunoglobulins. So remember, so this is kind of the summary here, where the B cells are circulating through the blood. So let's say it's a naive B cell. right? It comes in, remember the blood vessels flow up and through the lymph node tissue here. So here's just one of these capillaries that's cut in cross section. So the B cell traveled through that capillary and now it reaches this point in the capillary bed, which we call the high endothelial venule, special little region of teeny tiny veins, where the cells on the walls are expressing certain marks or certain markers that the B cell will sort of slow down and then actually squeeze its way out of the capillary and into the lymph node. So once it's in the lymph node, it may encounter matching antigen. For example, we learned that B cells typically are actually shown different antigens by big follicular dendritic cells that are hanging out in the primary follicle regions up here, right? And then that might activate the B cell, and at the same time, there will be T cells that are also activating against this particular antigen. Where's the antigen coming from? Well, it's being brought in. It's being brought into the lymph node from somewhere upstream, right? In this case, these lymph vessels that are coming in are the afferent lymphatic vessels heading into this lymph node, and they'd be bringing all kinds of fluid and debris, bits and pieces of cells, bits and pieces of pathogen, maybe entire pathogens themselves, like bacteria, are coming in. And of course, dendritic cells that are bringing things in to show off to other immune cells that are in this lymph node. So step 2 then, B cells and T cells will pair up, right? And then that T cell will encourage that B cell to make many copies of itself, many more copies, because up until now, this B cell is very rare, right? That it has by chance this particular shaped Ig on its surface that looks like it's going to be valuable, right? So your body doesn't want to lose that. So this B cell makes many copies of itself. And then working with the helper T cells, some of those B cells are encouraged to become plasma cells, right? And those plasma cells, then they start making the free-floating antibody version of the immunoglobulin, right? It just basically makes the antibody form of, for example, IgM as the first antibody that's released. But some of these B cell clones are encouraged by the T cells to go up into the primary follicle again, where they undergo hypermutation, right? So you end up with a big mass of rapidly dividing B cells, and they're doing that somatic hypermutation, trying to improve the antigen affinity. So every time the B cells divide, right, they have a slightly tweaked, mutated version of the Ig, and then even though the cartoon doesn't show it, you have the big follicular dendritic cells hanging out there, still showing these new daughter cells the same antigen, right? And so those daughter cells that bind tighter to the antigen receive more survival signals, right? And importantly, remember that those better B cells, they're coming back out and associating with the helper T cells and being made into the next wave of plasma cells. So now the antibodies that are being released from the lymph node are getting better and better over the course of the infection, right? Over the course of the 1 or 2 weeks that all of this stuff is taking place, right? And then lastly, as the infection is getting beaten and you're getting better, then your immune system switches from making more emphasis on effector cells that are going out and fighting the infection to keep you alive, right? Then the focus changes to encouraging more cells to become memory cells that are going to stick around for potentially months or even years to protect you against the reoccurrence or reinfection of the pathogen. Right. So then we shifted to chapter 7, development of T cells. Recall that B cells and T cells, they both start off from the hematopoietic stem cells in the bone marrow, right? And those stem cells, the hematopoietic stem cells, kind of go one of 2 directions. One is they can become the innate effector cells, or 2, they can become the adaptive effector cells, right? So this green lymphoid precursor cell is if the stem cell starts differentiating towards the adaptive side of our immune system. So this is a nice summary to remember that this yellow boundary here is meant to be, the bone marrow, right? That's why if you look in the cartoon here, the bone is yellow, right? So in there, B cells fully develop their immunoglobulins. So when the B cell leaves the bone marrow and goes into the blood, right, it already has immunoglobulins on its surface, and then it makes its way into secondary lymphoid tissues like the lymph node, like the spleen, or like things like the mucosal layer lymphoid tissues, where they might eventually become chosen to become plasma cells. And I always joke, right? I mean, the whole mission or goal in life of a B cell is to hopefully one day be chosen to become a plasma cell, right? And have that really important task of saving your life and protecting you. T cell, on the other hand, by the time it leaves the bone marrow, it still doesn't have any T cell receptor on its surface. We said it has to take a detour. From the bone marrow, it goes to the thymus, right? So this blue thing here is your thymus. So it's in the thymus, so this blue box here, that the T cell has to be encouraged to become a thymocyte that then starts trying to make a T cell receptor. Okay? And only after it's fully developed in the thymus and it has a T cell receptor, then is it ready to be let loose and go to work. Recall that the process of a precursor cell entering the thymus and becoming a full-blown T cell is pretty involved, right? First off, we say that this uncommitted progenitor cell that enters, this cell is really no different than the cell that left the bone marrow, right, and just made its way and happens to have found itself now in the thymus. But the tissues of the thymus will convince it through peer pressure. Right? Hey, you want to become a T cell, don't you? Right? Yeah, do something useful, become a T cell. So one of the important signaling events is this Notch1 signaling that convinces the uncommitted progenitor cell to become a thymocyte that starts doing the VDJ recombination. Right? So it starts expressing all the proteins that make the oven mitt, and then the oven mitt starts going to work. At first, the oven mitt tries to rearrange the beta chain locus, the gamma chain locus, and the delta chain locus at the same time. If it manages to make a good productive gamma chain and delta chain, then you sort of take a detour, go to the left here, and you end up making a gamma delta T cell, right? So the sort of brown and orange T cell receptor, right? One is gamma, one is delta. This is your gamma delta T cell, and it goes off and it performs sort of housekeeping types of jobs. It's more like it's detecting normal signals inside of tissues that might indicate that something is not right with some tissue that's old or damaged, whatever, right? However, if the oven mitt successfully makes the beta chain of the T cell receptor, then you're on your way to making an alpha beta T cell. And the alpha beta T cell is the kind of T cell we normally think of when we think about, oh, our T cells are part of our adaptive immune system, right? Because they're the ones that are going to be able to try to detect some novel antigen. So remember, at this point, the thymocyte begins to express both CD8 and CD4. These are those critical co-receptors that have to work together with the T cell receptor in order for the T cell to work, right? So at this point, it's a double positive because it doesn't know what it's going to end up eventually being, right? But then it tries to do the VDJ recombination again to try to make the alpha chain to go along with the beta chain. But at the same time, if in this process it makes the gamma and delta chains, then hey, this is your second opportunity to become a gamma delta T cell. And again, the gamma delta T cells do not have to undergo as much rigorous tolerance training as alpha beta T cells do. We will learn about why that is specifically in a later lecture after our exam is over. But for now, right, just remember, because gamma delta T cells are more concerned with general housekeeping types of things, the range of different signals that their T cell receptors are designed to detect is much more limited, right? And it's almost like they come with a more preset kind of set of shapes that they can use. Whereas the alpha beta T cells, right, they're all about random shapes, millions upon millions of different possible shapes. And so the alpha beta T cells, they have to go through additional training. Right? So at this point, if it makes an alpha beta T cell receptor, this green and yellow one, right, now, oh boy, now this is really going to be an alpha beta T cell, and then it has to decide, is it going to be CD8 positive or CD4 positive? Right? So I think one way of thinking about this is it's this multiple rounds of kind of positive as well as negative selection that goes into becoming a T cell, right? On the one hand, you're selecting for the T cells that are developing T cell receptor that can actually properly bind to things that they're supposed to bind to in order to work. And what I mean by that is, remember, a functional T cell only works by detecting small peptides that are presented to it by other cells, presented on things like MHC proteins that the other cell has on their surface, right? So in order for a T cell receptor to be able to actually function as a T cell receptor, you actually have to be able to have those associations between the T cell receptor and MHC proteins. So that's like a positive selection, right? As the T cell is developing, it actually has to have the right shaped TCR to associate with MHC. But on the other hand, right, you can't have that association be too tight, right? By chance, what if the T cell receptor is so perfectly the shape of detecting MHC that it doesn't even matter what peptide the MHC is carrying, right? That's not good either, right? So it's a lot of this negative and positive selection. And ultimately, remember, when the thymocyte has its T cell receptor, you will have dendritic cells in the thymus presenting MHC proteins to it, and whichever MHC type, MHC1 or MHC2, this T cell receptor binds to, that will select for whether it keeps the CD8 or the CD4 co-receptor, and that will then determine if this thymocyte becomes a cytotoxic T cell or a helper T cell. All right. Chapter 8 examined how T cells function to mediate immunity. We said that one of the important things to keep in mind is that a brand new T cell that comes out of the thymus, and remember, in the thymus, those T cells have to also undergo, the alpha beta T cells also have to undergo the self-tolerance training to make sure that their T cell receptors don't auto-react, right? So go back to your notes and refresh your memory on how that works. But now that this T cell is circulating around, right? It's naive, it has not bound to anything yet, and it makes its way to something like a lymph node. Right? The first time that it's presented some sort of peptide that it recognizes, right? So in this case, here's a dendritic cell, the yellow cell on its surface, it has MHC class 2, and it's presenting this red dot, right? This represents a small fragment of amino acids, a small peptide that comes from something that was processed inside of the dendritic cell, right, and it's showing it off. If this naive T cell, its T cell receptor happens to be the right shape to bind that combination of the red dot in the context of MHC, right, and then CD4 co-receptor, in this case, comes and confirms that this is MHC class 2, right? Together, that is like one signal, right? I mean, you can think of it as two signals, right? Because the T cell receptor itself is signaling and then the CD4 is signaling. But really, right, the CD4 is an integrated part of the T cell receptor complex working. Right? So that's one strong signal. But this naive T cell needs some confirmation that this red dot that's being shown to it is being presented to it by a trustworthy source, right? So in this case, this dendritic cell, because there is an infection going on, right? This dendritic cell is receiving different types of cytokine messages that encourage the dendritic cell to express this B7 protein, this dark green protein on its surface. And we said the B7 we think of as sort of like the salesperson badge, right? That allows you to know when you're sort of browsing around inside the store, if this person comes up and starts talking to you about a particular product, right? You're more inclined to listen to them, right? Because you realize, oh yeah, she works here, right? So hopefully she knows what she's talking about and she can help me, right? Compared to just some random person who comes up and starts talking to you, right? So in this case, the B7 signal is received by the naive T cell by its CD28 receptor on its surface. And that is your second strong signal that tells this naive T cell, hey, right, we are currently experiencing an infection right now. And this little red dot, we don't know what it is, but it's one of the many, many different peptides that we are currently processing right now. Is this something that you recognize, right? Because if this is something that you recognize, maybe it's really useful that you activate because this could be part, some evidence of the pathogen, right? Keep in mind, once this T cell activates and it goes off and starts doing its job, it no longer needs to see the B7 CD28 signal anymore. Right? I mean, that makes sense because, for example, if this were a cytotoxic T cell, right, you want to be very conservative about activating it. Right? You don't want it to activate if there's no infection going on. Right? But once it activates, because it sees the B7 CD28 signal from the dendritic cell, and once this cytotoxic T cell goes off and starts, targeting other cells in your body, I mean, those other cells in your body, the infected cells, they're not going to have B7 on them, right? Because remember, B7 is only expressed by a handful of professional antigen presenting cells in your immune system, right? So the B7 is used to wake up and activate T cells in a trustworthy way. But once those T cells are activated and they go off, they just need to see the red dot, right, being presented by other cells. Okay. Remember that helper T cells can differentiate into 5 different classes, subclasses of helper T cells, depending upon what type of coordinated effort is needed to fight against a particular infection. I always say, hey, a viral infection is different than a bacterial infection. It's different than a parasitic infection, right? In order for your immune system to have the greatest chance of successfully mounting an effective defense, it kind of has to know what immune cells need to be woken up and how they should talk to each other in order to coordinate an effective response. And I said that's the job of the CD4 helper cell. Remember, we used that example in class. If you have different disasters that are happening, like fire, biochemical toxic spill, or flooding, right? And you have something like FEMA, the Federal Emergency Management people, they hit the ground and they're trying to coordinate responses, right? You need to have people who are supervisors, who know how to communicate and organize people to deal with a flood evacuation or a hazardous waste cleanup, right? Or putting out a fire. So the CD4 T cells will differentiate into a particular subtype depending upon what's needed. And remember, how does it know what subtype to become? Again, this is happening inside the lymph node, and the lymph node is this environment that's sort of like a soup of different cytokine messages that are arriving from the site of infection. And those cytokine messages are like cries for help from the cells that are being damaged or killed at the site of infection, right? And that reveals what type of infection is going on. So in the lymph node, the CD4 T cells that are chosen and activated, right, they know what subclass to become. So be familiar with the 5 subclasses. You do not have to worry about the cytokines that trigger them to turn into these types, but you do need to know, right, that a Th1 cell, its job now is to activate macrophages, right? So for example, if there's a bacterial infection, this might make more sense, right? Th17, these helper T cells know how to talk to neutrophils to get the neutrophils working more efficiently. Th2 would mobilize certain antibodies in response to parasites, right? So for example, this would be the type that would encourage B cells to turn into plasma cells after they've class switched to make things like IgE, right? And then follicular helper cells, these T cells stick around inside the lymph node to help activate. They greet and activate more B cells that are coming in to make sure you have a strong, robust antibody response, right? Being able to tell B cells, okay, you become plasma cells, you go up into the germinal centers and work on improving your antibody fit, right? Recall that T cells, they carry out their jobs by associating or synapsing with other target cells, right? And then depending upon what that cell sort of presents that T cell will send different types of messages over to that target cell. These messages might be helpful messages. So for example, if it's a CD4 helper T cell that's synapsing with this B cell over here, and this B cell happens to have immunoglobulin that binds to the particular antigen of interest, right? The B cell's pulling these antigens in because it's constantly recycling the immunoglobulins on its surface. So as it pulls the immunoglobulins in, it also pulls in, chunks of, for example, the pathogen or whatever, and then the B cell is going to process that, and the little red dot represents just those small protein pieces from the chunk of the pathogen that it's bringing in. Right, so that's what presents to this helper T cell. The helper T cell likes what it sees, right? So it sends these green triangle cytokine messages back that encourage this B cell, hey, right, you should go ahead and divide, and then I'll help some of you become plasma cells. I'll help some of you go up into the germinal center and start working on your hypermutation. Right? Cytotoxic T cells, on the other hand, if they are at work and they bump into, let's say, a virus infected cell, right? In this case, hey, there's still the red dot in here, but the red dot is part of this cell, not because, like the B cell, not because the B cell is doing its job and binding to this thing and bringing it in and showing it off. But in this case, this yellow cell down at the bottom, hey, this one became infected, right? So as a result, the little red dots get loaded onto MHC1, right, compared to the MHC2 that the B cell uses up here, right, to pass the message on. So the cytotoxic T cell will see the red dot on MHC1, right? That's what its CD8 co-receptor confirms. And then the messages that the cytotoxic T cell sends over, right, are actually messages that tell the virus-infected cell, you better undergo apoptosis, right? You better self-destruct and take yourself out of the picture before you end up releasing a whole bunch of new virus particles. Okay, chapter 9 was talking about how B cells and antibodies work. So this figure, I think, is a really useful review figure. It's busy, but if you're using it for review, you can take your time and just kind of like digest it, right? So what we're saying is, if you have two different infection scenarios here there's a virus that infected tissue, here is a large parasite, right? Keep in mind, the virus is teeny tiny, right? A virus is way smaller than, let's say, your typical dendritic cell or neutrophil, right? Whereas something like a parasite might be considerably larger than your typical immune cell, right? But they're both cases of pathogens, and obviously they have to be dealt with very differently. In both cases, you have evidence of infection draining into things like your lymphatic system, right? So here there are little pieces of virus particle or other cytokines, right? Similar down here. And in this lymph node, you will have things like cytotoxic T cells. So these were CD8 positive T cells that then wandered in here and then activated, right? So they saw not only the dot, the peptide that their T cell receptor recognizes, but it was shown to them by some type of immune cell that is also wearing the B7 salesperson badge, right? So the cytotoxic T cell, it activates and it leaves and it goes elsewhere in the body, hoping to bump into infected cells, in which case they will send their cytotoxic messages and tell the infected cells to die. Right? So the CD8 cytotoxic T cells, they kind of have a one-track mind, right? They're pretty straightforward because their job is to just go out there, bump into cells that are infected and tell those cells to die as a way to try to slow down the spread of something like virus. Right? The helper T cell is a bit more complicated, right? Because the activated helper T cell, it kind of has to pay attention to the cytokine soup that's going on in here, telling it what the nature of the infection is, right? Is it a parasitic infection? Is it a viral infection? Is it a bacterial infection? And based upon that, it will go ahead and turn into one of the 5 different subtypes or subclasses of helper T cell. Right? So then this helper T cell will go ahead and associate with B cells. And so remember, this helper T cell has to see the peptide that it likes being presented by the B cell. That's how it knows that this T cell and this B cell are sort of like meant for each other because they can work together. Right? Remember also that the activated helper T cell expresses CD40 ligand on its surface, and the B cell has CD40 receptor. So this is the way that this T cell alerts this B cell that, hey, I'm about to send out some useful messages here, and these are meant for you, right? Not meant for the other B cells that are floating around nearby that have completely different immunoglobulins on their surface, right? You don't want those to activate because that's like a real waste of time, right? So this B cell, because of this CD40 ligand, CD40 receptor signaling, knows that it pays attention to these different cytokine signals, and it will then tell the B cell what to do, right? Maybe it'll turn into a plasma cell and release IgM. Remember, the IgM floats around as these pentameric rings. Or maybe it tells the B cell to class switch, right? For example, if it's a large parasitic worm that your body's dealing with, maybe the B cell needs to make IgE, right? Because IgE can then float to the site of the infection, and you have granulocytes like mast cells, basophils, eosinophils that have Fc epsilon receptor on their surface. They can grab onto the stems of these IgEs, right? And once they get suited up, remember that's like the Tony Stark getting suited up as Iron Man, right? This sort of mild-mannered granulocyte that normally is kind of an innate effector, right? It has no targeting ability. It just kind of wanders around until something triggers it, and then it like poof, releases all these types of noxious, nasty histamines and things like that. But when it's suited up with IgEs that are specific to, let's say, this parasite, then all these granulocytes can stick to the parasite, right? Because they're coated with IgEs that detect different signals on the parasite. And then when they release their histamines and other noxious kinds of things, right, they can much more effectively attack and irritate the parasite, right? Or they might turn into IgGs, right? Or it might be that the plasma cells are told to make IgAs. For example, if it's a viral infection. If it's a viral infection, you don't need to make IgEs, right? You need to make IgAs that can then fill up your mucosal layer, right? Because the IgAs form these little dimers, these little bow ties that, remember, are very, very abundant in your mucus layers to fight against things like virus particles that you are breathing in. All right, so the intersection of B cell and T cell activation, very important, right? Very important that you understand this, because this is one of the most powerful, important events of your adaptive immune system waking up and mounting a strong defense. Because after all, right, B cells make antibodies, right? Antibodies are very effective, but B cells don't know what kind of antibody to make, or that they even should turn into plasma cells and make antibodies unless they're told by a T cell what to do, right? So you have to have B cells and T cells working together, paired up, right? So having a B cell that happens to have the right immunoglobulin that can attach to some sort of pathogenic fragment or chunk of something that comes from the pathogen, so that the B cell can present the little peptide pieces of that for this particular helper T cell to pair up with, right? And then working together, then you're really in business, right? Because as a pair, remember they do this like kind of little dance here, the T cells will escort the B cells up into the germinal center, and they'll also escort the B cells to become plasma cells, right? So very important that they both end up recognizing the same signal. And remember, right, the immunoglobulins on a B cell, they are not limited to binding to just peptide, right? They can bind carbohydrates, lipids, whatever, right? But the important thing is when they bind to that, chances are it's a chunk of something that's not just carbohydrate, but has some lipids in it and has some protein in it. So when the B cell takes that in and processes it, right, the little red dot is just the peptide parts that come out. That's what it presents. So it's able to communicate with a helper T cell that recognizes the red dot. Right. Chapter 10 dealt with mucosal surfaces. So remember, the majority of the surface area of our body is not what we usually think of as being like the surface area, like our outer skin, right? The majority is actually the layers of mucosa that cover things like our respiratory tract, our gastrointestinal tract, our urogenital tract, etc., right? Substantially more surface area there. And remember, it's particularly challenging because not only do you have way more surface area to deal with, but the mucosal layers by design are very thin, very fragile, right? Why? Because those are the surfaces that things are moving from the outside to the inside, things like gases, things like nutrients, right? And vice versa, waste products, CO2, right? So these mucosal layers are very thin and delicate, And also, ironically, right, that's where the majority of microbes that we're carrying around can be found, pressed up against these fragile layers of our respiratory tract and our gastrointestinal tract, especially our gastrointestinal tract, right? We talked about just like the thriving communities of was a commensal bacteria and microbes that we evolved to actually expect to be part of our digestive tract because they help to break things down and digest things and help to keep bad microbe populations, in check, right? So recall that one of the key defining differences between how our immune system deals with a systemic infection, so for example, a cut in your skin on your arm, for the first part of the semester, we learned about that. We said, oh, you have macrophages there, right? And they start going around eating up the bacteria, and the macrophages start releasing cytokine messages to try to encourage inflammation, right? Meanwhile, you also have tons of complement protein in there, so they're all working together, right? If your immediate response is not enough, so after a few hours, maybe a day, you still have a lot of bacteria there. By then, the cytokine levels are building up, and the interleukin, the inflammatory cytokine levels are building up, and you start getting inflammation, right? And the inflammation then is kind of damaging, right? It causes your tissue to get damaged. The capillaries get leaky so that more immune cells and more complement proteins can leak out in there. And as a result, when the infection is done, you have some tissue damage, right? You have some scab, scarring, maybe things like that. But it's okay. I mean, because, it's better than you dying from it, right? You have to live with a little scar. We said that the mucosal response is very different, right? Because when you're dealing with the really thin, really delicate layers of your mucosal layer that have tons and tons and tons of microbes pushed up against them, the last thing you want to do is make things way worse by causing inflammation that might allow, right, way more microbes to gain entrance if you start damaging your own mucosal layer. So as a result, our mucosal immune system, right, really tries to avoid encouraging any additional inflammation from happening. Right? There may be some inflammation that's just a direct result of microbes coming into your tissue and damaging your tissues, right? And therefore your cells that are getting infected might yell for help in the form of cytokine messages. But the way your immune system responds is it tries to suppress any inflammation. Right? So those immune cells that are part of our mucosal lymphatic system, and remember, right, because we're talking about a large surface area, it is all surrounded by secondary lymphoid tissue, what we call the MALT, or mucosal associated lymphoid tissue. And the MALT that lines our gastrointestinal tract, it has a particular set of challenges, right? So we even call it the GALT, right? The GI tract associated lymphoid tissue. So GALT is like a type of MALT, right? And so the cells there that react, right? You have way more immune cells there present to start with, right? So most of your immune cells are actually located in your mucosal layers right now, right? Ready to deal with potential infection. And so they act a lot faster, and importantly, they don't release inflammatory cytokines, right? So that hopefully the infection can just be resolved without any additional damage to your tissues. So importantly, oral tolerance. We said that our intestines, especially our small intestines, are constantly sampling what's going on, what's passing through them in the lumen. That's the way our immune system sort of keeps up with what's going on, like different populations of bacteria, as well as any bacteria that you are unintentionally eating with your food, right? Every bite of food, there's some bacteria that you're eating and viruses, right? So we said, how does your body, your immune system tell the difference, right? As you're breaking down the meal that you're eating, and you have many, many different potential small peptide fragments or antigens that are just part of your burger, fries, or soda, right? How does it know that those dots, right, are not a threat? But at the same time, there's lots of bits of antigens coming in from bacteria and things that are hitching a ride on your food, right? And it all gets sampled. How does it know not to freak out against things like food antigens, right? And again, It all has to do with the lack of inflammation, right? If you eat a normal meal, right, and you're totally fine in the course of your normal day, and there is no evidence of infection or inflammation, right, your mucosal lymphatic system will just say, hey, you know what, everything seems to be fine, right? And that includes going to the extent of any naive T cells, for example, that are suddenly binding to something that a dendritic cell shows it, right? Your immune system plays it safe. And it says, you know what? There's no evidence of infection going on. We're just eating lunch. So I'm sorry, but even though your T cell receptor bound to something here, you know, without any compelling evidence that we need to actually activate you, you're better off just becoming anergic so you don't cause problems. Right? Okay. So when you do have T cells activating, right? So remember we said that importantly, T cells that activate and B cells that activate as part of something like a Payer's patch that's sampling food if there is an infection going on, right? And they detect that these particular lymphocytes are the ones that know how to recognize them. They will drain into the mesenteric lymph nodes that are part of the big mesentery. That's the big membrane system that kind of holds our guts together, right? Keeps our intestines positioned properly. And importantly, then eventually those activated cells will make their way through our lymphatic system, get back into our blood, right, close to the heart, and the heart will then pump it throughout our body through the blood. And as I told you, those activated cells won't just wait to get back to, for example, your intestines, right? They will start spreading out in many of the mucosal layers, for example, your respiratory tract, just in case, right? Whatever it is that you ate is also something that you might be breathing in, right? So those plasma cells that then make IgA, and you have those T cells that will actually squeeze their way and place themselves in the epithelium of your mucosal layer to serve as little sentries, little guards, right? Okay. The last chapter has to do with immunological memory and vaccines. Right? Oh, I didn't realize it's 11:20 already. Sorry. Right. So remember, memory is a very critical part of our adaptive immunity. Right. So we said that the first time you're exposed to something novel, like the googly-eyed drooly virus, you know, your innate response will have first crack at it, right? But the point is, if your innate response cannot deal with it, then after a week or so, you start having to really start depending upon waking up your adaptive immune system, right? So you'll have a big spike in the amount of effector T cells that are selected and activated, and a big spike in selecting for B cells that can then produce large amounts of antibody. Right? So even though maybe by the end of the second or third week, you feel a lot better, right? And you're no longer stuck in bed, you know, hating life and feeling bad for yourself, you go back to work or school, your adaptive immune system is still pumping out high levels of antibody because you go back to work or school, and guess what? You're repeatedly exposed to the googly-eyed pathogen again, right? Second, third time, whatever, because that's where you caught it from, but you're protected now, right? Because the antibodies right away surround the pathogen and neutralize it. Then for months or years, you have a certain level of memory B cells and T cells that are sticking around, and you have this elevated level of antibody, right? Specific antibody that represents very good quality antibody because it's the last antibodies that your adaptive immune system figured out how to make in the course of B cells doing the hypermutation, right? So they stopped here and then just hung out as memory cells. So the next time, months or years later, googly-eyed drooly pathogen comes back, right away, well, you have already high quality antibody in your system. Maybe that's enough, right, to help cover the virus so that complement proteins can cover it much faster, so that macrophages can eat them up much faster. So your secondary infection here, right, with the help of antibodies, your innate system working together with antibodies help them out, hopefully is enough to neutralize the pathogen, and you don't even get sick, right? But your adaptive immune system, boy, it really wakes up in the background. Why? Because now it knows, right, that this googly-eyed pathogen is something that's continuously out there, right? And that you're going to be exposed to it again and again and again. So now it knows, hey, we should take this seriously, right? We were infected with it once years ago, but we didn't know if we were ever going to see it again. So we weren't really sure how strong of a immune response, immune memory to have. But now that we know that it's something that came back, right, you have a much stronger secondary response. And remember, with each additional exposure to that antigenic signal, not only do the levels of steady state antibodies as part of your memory go up and up and up and stay up, but the quality of your antibodies improve, right? Because whatever B cells, whatever cookie recipe they had the last time that they saved your life, that becomes the starting point for further hypermutation the next time you're exposed, right? So we said, that's why certain types of vaccinations, like the COVID vaccination, you get 2 shots with the Pfizer and the Moderna type, right? The first one starts building up your adaptive immune system, wakes it up, right? And you have some memory of it, but it's the second follow-up shot that really gets your immune system to realize, hey, this is something that we're going to see over and over again. So it's worth it to invest in making really, really strong immune memory. And that's why, right, it's your second shot that oftentimes makes you feel a little sick, because as you have this huge immune response, you have some background inflammatory types of responses, and you might feel achy or tired or whatever, right, as your immune system pays more attention to it following your second shot. Right. This table, go back and review. I'm not going to go over it now, but this is just some differences between the primary response, the first time that you're exposed to the googly-eyed drooly pathogen, right, and the second time you're exposed to it, right? So think about what this has to do with what we just were discussing in terms of immune memory and your immune system being encouraged to take things seriously. Right? And we said that the concept of vaccines is based upon the fact that just as long as you expose your immune system to whatever, you know, 3-dimensional shaped molecules or whatever that you want your immune system to learn, right, as certain signals as being from the pathogen, so that you can select for things like antibodies, right? If you can present those things to your immune system without presenting the actual virus or the actual pathogen itself, then this provides you a way to give your immune system a heads up, a sneak peek, to get it to start building immune memory, right, without any risk of you actually getting sick from the virus. So Mother Nature, you know, never sort of intended it that way, right? Mother Nature intends you have to get sick from the virus the first time in order to see all those potentially good antigenic signals, and then you select for B cells, right, and T cells that recognize them. But, you know, you might die and not even survive that first infection, right? So vaccines are when you just take basically the particular antigens and you allow your immune system to see them, sort of like simulating or a mock infection, right? So that your adaptive system gets to build up that immune memory, even though you never are exposed to the full virus, right? So that could either be a lazy form of the virus, or with the COVID vaccine, you know, mRNAs that actually make just like the googly eyes, right, of the virus, so that your cells in your body present those to your immune system. But either way, right, your immune memory that you build up protects you, so that when the real mean, aggressive googly eye virus comes to you, right, the real COVID virus comes to you, you already have the immune memory to protect you against it. And then this was just a summary, right, that there are different strategies for making vaccines. You can use the intact killed virus particles, but that's not really that great, because your immune system, you know, it might not really care. These dead virus particles that don't do anything, they're not going to elicit any kind of natural, real infection-like response, right? Other vaccines come from the viral proteins that are used. Some vaccines, you can actually use bacteria to try to trigger your immune system. And in some cases, right, depending upon what is in the vaccine, you might need to have, as part of the formula of the vaccine, certain adjuvants or things that are added to the vaccine in order to really stimulate your immune system and get it to pay attention. So these could be pieces of bacterial proteins, lipids, other chemicals, or certain types of metals like aluminum, because we've found that those types of things stimulate inflammation, right? And once you're stimulating inflammation, that's part of really getting your immune response to believe that there's an infection going on, right? And then we discussed at length why the Pfizer and Moderna mRNA vaccines are particularly exciting, right? Because they actually just make your cells produce the actual antigens on their surface in a way that looks very convincing to your immune system that you are experiencing infection. Okay. Okay, so that's it for the review. Let me stop the share here. Let me stop the sharing here. Okay, so I'm happy to stick around if any of you have any questions. Just type your questions in the chat. Don't speak up, because what I'll do is I'll just record these questions this time also. So I need to kind of keep it anonymous, right? Who's asking the questions? All right, so in the chat, here's a question. What if the actual lymph node is infected? Right? How does it let everyone know? Same way as any other cell? Yeah, right? If the infection is actually taking place inside the lymph tissue itself, then yeah, I mean, it's no different than if the cytokine messages were coming somewhere else, right? It would be the same types of cytokine messages. And so that environment in which the B cells and T cells are activating, is even more, I guess, direct and immediate. Here's a question. Is the T regulatory cell highly activated in the gut or other mucous membranes to try and dampen down the excitement of T cell response to incoming bacterial fragments from food? Yeah, so we have elevated Treg activity in places where it's more likely that we will be exposed to novel antigenic signals coming from things like food, right? I can't emphasize enough how important that concept is and how interesting it actually is, right? Because remember, I told you, if you think about in our typical modern diets, right, we eat so many different types of foods that are way more variety than I think Mother Nature sort of designed us to be able to sort of normally anticipate, right? I always think about, if we were still just living in our little communal groups, like a little village somewhere, and every day we only typically wander, at most 5-7 miles in a radius around our village to maybe hunt for food or forage for food. Most of the time we'd just be back in the village, maybe growing food, right? The different types of antigens that we are normally exposed to in the course of our daily lives is much more limited than what we experience now in our modern society, right? Where you have all this processed food and anything you feel like eating, you have a favorite restaurant that you can go to drive through, whatever, and get it. Right? So how is it that our immune system doesn't freak out at all these strange different antigens that are being sort of absorbed by our body, right? It has to do with suppressing inflammation. So if you eat a meal and there is no real bacterial infection going on, right? Your immune system tries to keep all of your immune cells from freaking out, right? And part of that is the activity of these Treg cells. As we will learn later, Treg cells play a very important role in the development of certain types of cancer. Okay, so we end this semester with a discussion of cancer because, not surprisingly, the immune system plays a very important role in the development of cancer. All of my immunology training actually comes from the fact that I'm trained as an oncologist, right? I'm a cancer researcher. And so all my immunology knowledge comes from understanding the development of cancer. But we'll get to that. So Tregs can play a very important role in interfering with not only our immune system properly, ignoring antigens that are coming in from our food that look weird and different, but in some cases, Tregs, if they get out of control, can actually suppress our immune system from activating when we want it to activate against things like cancerous cells and things like that. All right. Next question. All right, kind of an odd question. All right, so I'm just going to read on here, right? Something tells me I should probably preview this first. Kind of an odd question, but I recently saw a video on TikTok. Oh boy, this is going to be good. That was explaining how a woman's immune system immediately recognizes and starts attacking a male sperm It made me think, would the repeated attempts to try to become pregnant progressively become more difficult because the woman's immune system will gradually create better immune cells with higher affinity antibodies? Um, I don't necessarily think this is true, okay? Because remember, we make a very big deal in this class about understanding what's inside versus outside of our bodies, right? Even to the extent of saying, you put food in your mouth and you swallow it, the food goes all the way through your system and it's still outside of your body, right? It's just like a car traveling through a tunnel through a mountain, right? The car's never really inside the mountain, it's passing through the mountain. But we said that's true of our respiratory tract, and we said it's true of our urogenital tract, right? So so the space inside, for example, the uterus, or even inside the fallopian tubes, that is still technically outside of the body. All right. So while, yes, right, those are considered mucosal layers as well, so you'll have a lot of IgAs, right? You're not going to have strong immune responses against things that are still outside of the body, right? So I'm not sure what this TikTok video was. I don't think I'm going to go to a lot of trouble trying to find it, because after all, it's a TikTok video, right? So it'd be kind of bad if your immunology professor was basing what I'm teaching you on TikTok videos. So I don't think that's really a big issue. Okay. If you want to, and you find some more information about that, forward it to me, because I'd like to see what they were talking about. Okay. And think about it this way, quite frankly, evolutionarily, that wouldn't make much sense, right? Because if you had immune systems that had heightened ability or sensitivity to have a woman developing immune responses that reject things like sperm, right? I don't think that would actually be selected for by natural selection, right? Okay. How does your immune system know to let commensal bacteria hang out versus mounting an attack, right? Again, it's because the commensal bacteria is normally outside of our body, right? So in the lumen of your gut, in your small intestines, that's still outside of your body. Commensal bacteria, right, that's where they should belong, right? If commensal bacteria breach the wall of your small intestine or large intestine and actually get into your tissues, they're no longer considered commensal bacteria, right? Because those bacteria, I think I said this before, I don't remember if I did or not, but I often joke that the good commensal bacteria that's hanging out in your gut, right? If you end up with like a lesion, like a cut in the wall of your intestines, maybe from something like a ulcer, right? Something like that. Because, ulcers that are often caused by things like stress causing stomach acid to weaken the mucosal layer. And then you can get a lot of inflammation and pain, right? Where does that come from? Well, that comes from the fact that you have commensal bacteria that's then breaching the wall of your tissue and now is going into your body, right? And it's not like the commensal bacteria, like the good E. coli, once they're in your tissues, suddenly stop and go like, whoa, right? I'm not supposed to be here. And then they run back into the lumen. No, right? They're going to be like, wow, this is great. I didn't know that this was here. I'm going to go ahead and go crazy now, right? So the moment that happens and they enter your tissues, they stop becoming good commensal bacteria and they become pathogenic bacteria, right? So as long as they're still outside of your body in the lumen of your intestines, right, they're separated from your tissues by the mucosal layer. So yeah, if any of those bacteria make their way into your mucosal layer and they're met by things like IgAs, right, yeah, those bacteria might be destroyed, right? And that's one of the ways your body keeps the good bacteria out from getting into your tissues, right? But allows them to still thrive inside of the space of your lumen in your intestines. Okay. Hopefully that answered your question. Oh, am I going to continue my cliffhanger story from before spring break? Yes, right. I think I mentioned last lecture, I left you guys kind of hanging with the story about me and the soy caramel macchiato from Starbucks when I was a postdoc in Boston, and how I felt like I was about to die and kind of made an ass of myself in front of my PI. I was all embarrassed and laughing, even though I was actually kind of scared. But anyhow, I realized that at the time I was telling the story, I forgot that like we have this second exam coming up so soon after spring break, right? So I realized, oh, it's better that I put off the ending of my story until after this exam, so we can just focus on getting you guys through this exam first. And besides, it turns out that the ending of my story fits a little better with our discussion about things like food allergies that we will talk about, in the final part of our class, right? After this exam, we'll start talking about stuff like that. Okay. So yeah, sorry to keep you hanging, but we'll finish that story at a time when I think it'll actually make a lot more sense to you. All right. So that's it for the questions. I'm going to stop the recording and call it a day.